So how many experiments have you done that are as a function of something, a function of temperature, a function of concentration, a function of pH, and yet, which tells us what? That environment matters, right? And yet, what are you studying under those conditions? Something that's been fully extracted, likely, from its native environment. Protein, for instance, which was evolved to function in a membrane, in a whole cell. And yet, over and over and over again, techniques like UV-Vis spectroscopy are being used to study proteins in a clear buffer. And why is that? Well, part of it is the limitations of the UV-Vis spectrophotometers themselves. If one has particulates in the solution, if it turns into a suspension, the light scatters. So instead of light traveling straight through a sample to an awaiting detector, it gets scattered all over the place. And so the detector only sees some percentage, often a very, very tiny percentage of what should have been part of the absorbance light measured. So one gets the wrong answer. And so you go back to the bench and you clarify more, you purify more, you centrifuge, whatever. You dilute, you do what you can in order to minimize scatter. Well, while there are existing technologies to help you around that, and I'm thinking of diffuse reflectance in particular, diffuse reflectance is a reflectance measurement, not an absorbance measurement. The only way you can truly work with a suspension, including whole cells and proteins in them, is with a technique called, or a little device called, an integrating cavity. So instead of this light scattering all over the place outside of a cuvette, light is contained inside of an integrating cavity, so that the only way the light escapes is through an intentional port directly to the detector. So this was developed about 10 years ago, 12 years ago or so, and there's a number of fabulous publications in the literature showing the importance of working with systems, proteins and everything else, not in a clear buffer, but in a salty environment, in adjuvants, in vaccine development, in a whole cell, in whole tissue. These experiments have been done. So if you'd like to learn more, please be in touch.